Okay, so now what we've done is we've said, okay, we have materials inside of our body that can change the excitation state of other elements. And we can do this by increasing the temperature, the concentration, maybe change the pH of range of something. But most importantly, there has to be some form of a solvent around to allow these things to actually mix with one another. That's ultimately what you should have taken away from the first two sets of presentations. Okay. Now what we're going to do is we're going to look at carbon. And the reason why we do this is because carbon can actually attach many functional groups to its essentially structure. So what I mean by that is, here's carbon. And I always use the analogy of Lego blocks, but some people don't like Lego blocks, and I have to wonder what's wrong with them. Okay? And if you fulfill the octet rule, you know that there are 4 valence electrons. Let's put a dot here. There's 4 valence electrons around a carbon at any given point in time, because the 2 are filling the innermost shell, and that means Mother Nature can attach a functional group at any one of these sites. Okay? Think about changing the blade on a reciprocal saw. Pretty much the same thing. You put a different blade on it, it does a different type of cutting. In this case, what happens is Mother Nature can attach either an ester, an amino, a carboxyl, a phosphate group to any one of these, and now you've changed the chemistry of that carbon, allowing it to enter into many different types of chemical reactions. But the basic 4 again are anabolic or synthetic, catabolic or hydrolysis based. You can have an exchange reaction, or you can have a reversible reaction. Okay, so having said that, isomers of the same molecular formula but different structures. So here's where students really get lost. C6H12O6, you're just counting carbons, hydrogens, and oxygens here, and that could be glucose, but it could also be fructose. And because it could be either of these things, and when you actually look at the physical structure of it, we call them isomers because they're not the same. We don't spend a lot of time thinking about this, but why would Mother Nature do that? Mother Nature would do that because there's going to be a specific receptor for glucose or a transporter for that. And that transporter won't let fructose in. Okay? So the shape and its charge, because of those functional groups, determine what happened to those molecules. So it's important for us to understand that there are isomers. Okay, so the first set of structures we're going to build are carbohydrates, right? So we've got to get rid of that bad carbon drawing for you there. And carbohydrates provide most of the energy needed for life and include sugars, starches, glycogen, and cellulose, right? Well, like, yeah, okay. Provide most of the energy needed for, we should actually qualify this, short term or fast energy needed for life that includes sugars, starches, glycogen, and cellulose, because, hey, sugars, it's like a piece of paper on an open fire. Starches, a couple of pieces of paper. Maybe a small book, all right? We don't break down cellulose. In fact, we use it as an irritant. So when you're looking at carbohydrates, you want to be thinking about the structural complexity of those carbohydrates, because carbohydrates are divided into 3 major groups: monosaccharides, okay, disaccharides, and polysaccharides. And depending on how we actually assemble them, let's say we have polysaccharide here. Well, that's glycogen. That's polymers of a simple sugar, glucose. Okay, so Mother Nature has something going on here, where by increasing complexity, she can actually store energy inside of those shapes. So what are the shapes? Well, we have pentose sugars, which are monosaccharides, where we have deoxyribose and ribose. And that should sound familiar because deoxyribose sugar is the backbone of DNA. Ribose sugar is the backbone for RNA. And they're pentoses, but because they're single monomers, they're also monosaccharides. Now let's take a look at the hexoses. Notice hex is for 6. Try and count the 6 carbons on this. If you know how to do that, you're doing better than most on the face of the planet. And if you don't, you should ask that question in class. Glucose here, notice it has a specific shape, and notice it has a specific color, and that's why it's called a hexose. But you're looking, hey, fructose doesn't look like this, but it's still C6H12O6. Go count the carbons, prove it to yourself. Okay, and you're going to see this is what I mean by a specific shape will allow it to be transported into the cell. A specific shape will not allow it to be transported into the cell. Okay. Disaccharides. Well, this is when we put them together. And now look at the vocabulary change. I don't really care if you know that glucose and fructose gives us sucrose. Yay, big effing deal. Okay? What I do hope that you see is that this is a dehydration synthesis reaction, because look at what's happening here in green. We're taking a hydrogen from here and a hydroxide from here. We're removing them as we are synthesizing them to make sucrose. Now we have sucrose and water, and that's why it's called a dehydration synthesis reaction. And it's called a hydrolysis reaction if we're having it go in reverse. So this would make this a reversible reaction. We now add the water. It's hydrolyzed. It's broken into H plus OH, and then they are added to the actual sites they were taken off of. Okay. You don't need to memorize the example I just went through. What you need to understand is, what is a dehydration synthesis reaction? What would your products be? Well, it'll be one product plus water all the time. If we need hydrolysis reactions to occur, you need to have water in the body. So if you're dehydrated, the rate of hydrolysis reactions for you to break materials down is going to go down. Polysaccharides, the one that I gave you, of course, is glycogen, and it's stored inside the liver and or skeletal muscle tissue. Okay. I don't know why they say or there, because it's in liver and skeletal muscle tissue. Okay, and when your blood sugar level drops, your liver is going to break that down. That's why they call it hydrolyzing it. It's a hydrolysis reaction to give you individual monomers of glucose, which will enter into your body. Now, lipids, which is where we're moving to next, like carbohydrates, contain carbon, hydrogen, and oxygen, but they do not have the 2-to-1 ratio of hydrogen to carbon. Okay, they actually have fewer polar covalent bonds, and they're hydrophobic. Okay, they're mostly insoluble inside water, and in those instances inside chapter 18 in your future, when you need to understand how lipid-based or steroid-based hormones travel inside your body, they have to actually combine with a protein. Hence, they're called lipoproteins, okay, because they are now allowing these lipids to become soluble inside of the circulatory system, because normally they wouldn't be. Here are two examples of fatty acids. These are lipids, okay, and this is palmitic acid. So lipids are fatty acids, or fatty acids are lipids. And in this instance, it's C15H31, C double bond OH, right? And what you actually have here, C double bond OOH, okay? What I want you to see here is this is a saturated linear structure, and this is an unsaturated nonlinear structure. These are more healthy for you because they create space. Why do we care about these? Well, we need them to make triglycerides, which are the most plentiful lipids inside the body. They provide protection, insulation, and energy for both intermediate energy uses as well as stored energy uses. And the significance here, of course, is they can provide twice as much energy per gram as either carbohydrates or proteins inside of your body. So that's why we store them this way. Anything that you have inside of excess inside of your body for either carbohydrates, protein, fats, oils, it's all converted into something, because Mother Nature doesn't know when you're going to eat next. So what she does is anything that you had extra of, that last bite of, I don't know, Ben and Jerry's Cherry Garcia, right? You told yourself you weren't going to eat the entire pint, but you did, okay? And whatever your body didn't need, it converted it to triglycerides, because it doesn't know when you're going to be able to do that again. And what it's doing is it's measuring how much energy you're taking in and saying, well, if you can provide me this much, I'm going to expect that much. And if you don't give me that much again, well, then I've got a problem, and I need to start storing it, okay, until we start breaking it down. How do we build these? Well, we have these things that are chemical reactions between each of the fatty acids and, in this case, a glycerol backbone. And what's going to happen is we're going to undergo a series of dehydration synthesis reactions, 1, 2, and 3. And you know that because we have one molecule of water, second molecule of water, and a third molecule of water here. So you'd end up with a triglyceride. Where did you go? A triglyceride that has now an ester linkage between the glycerol backbone and each of those fatty acids and 3 molecules of water. You can do the algebra. If I had you do 10 of these, you could imagine 3 formations of triglycerides. You'd end up with 30 molecules of water. Okay, don't worry, I won't be doing that. But I just need you to think about it. Now, phospholipids are an important membrane component, right? And there's lots of reasons for the textbook to sort of like drop this in on top of you like a book. But the reality is I'm hoping what they were thinking is, okay, so we've given you the basic building blocks. We gave you some carbohydrates. Now we have some fats. Well, what's the consequence of actually having a fat that we can attach a phosphate to? Well, the consequence is we can actually make membranes. And that's what I'm hoping they're talking about here, because that's the way I would do it. Okay? They're making membranes. And remember, these are eukaryotic cells, and therefore membranes allow us to create partitions so that chemical reactions can happen between those partitions. The interesting thing about phospholipids that make up membranes is they are amphipathic, which is a big fancy word for saying that they are both polar and nonpolar, and you'll see how that happens in a moment when I actually show you this. Well, here's the polar head and has a phosphate group inside it, which is negatively charged, which means water is going to want to interact with this. And it has nonpolar tails, and in this case it looks like we have monounsaturated here and a saturated fatty acid here. But what's significant is water doesn't want to interact with this here. So what Mother Nature did is she put the polar heads here, and put nonpolar tails, tail to tail. And in doing so created an impermeable membrane to water. And it's not completely impermeable. Every time these things move, it's not like they're stationary. Maybe a water molecule gets in there, it gets pushed to the other side. But not enough to make it useful to you. Okay? But however, at the end of the day, you now know we need to have a glycerol backbone with a phosphate group attached to it and two fatty acid tails to make up a phospholipid, which will help us make membranes. See what I did there? Okay, cool. I'm hoping you did. Now, lipids can take many different forms. Steroids have a specific structure to them. They have 4 carbon rings. We use them as bile salts, some vitamins. Cholesterol is the major building block for synthesizing steroids. And that's primarily inside the smooth endoplasmic reticulum, inside of your liver cells. Okay? And what I want to do is I want to show you just essentially what's going to happen to them. Well, here's cholesterol. 1, 2, 3, 4. 4 sterol ring structure. And because this is cholesterol, it has a hydroxyl group here and a hydrocarbon tail here. We don't tell you what it is. But now what I'd want you to do, if you had the time, is compare this to each of the 4 different molecules here. And you'll see that, generally speaking, they're similar. They're similar in that they are made up of a lipid, that is a sterol structure made up of 4 cyclical structures. But then we change the functional groups and how hydrated they are. You'll notice this double bond here is shared here, but you have 3 double bonds here for estradiol versus testosterone. Okay? And that's what Mother Nature's doing. She's undergoing a series of reactions, making these subtle changes that don't look all that significant to us, but because you've changed the shape, you've changed the charge distribution, and now you've changed the function of this molecule. Okay, let's go off to proteins, okay? And I think I'm just going to try and muscle all the way through this. Yeah. So here's proteins, okay? Constructed of 20 different amino acids. What we do is, of course, here's the backbone. An amino base, a carboxy acid tail group. We have a side chain. That R means we can add whatever we want onto this. Think of it as a Lego building block again. And you'll see that there's a hydrogen here for all of them. Look at them, like you're like 5 or 6 years old. Notice we don't give you all 20 amino acids inside AMP1. They save that for your biochemistry classes, although you need it for AMP2, which is kind of interesting. How do we actually make peptides? Well, if here's an initial amino acid and we need to make, say, glycylalanine here, which is a dipeptide, and this looks very much like the earlier chemical reaction we saw. We have a dehydration synthesis reaction, and now we have a water molecule. So you know what happened. We took a hydrogen here, we took a hydroxide group here, hydroxyl group here, and now if we wanted to break this down, like imagine you had protein for breakfast this morning, and it had that dipeptide inside of it, guess what? You're going to hydrolyze it. Well, yay. We have 20 of these. You can imagine the complexity between polypeptide chains contain between 2 and 20,000. Okay, some of the numbers your textbook has are just plain wrong. Okay. We need to organize this, though, because for you to be alive, you have to be in the correct tertiary shape. We need to understand what tertiary shape is, okay? And that means we actually have to build a primary, secondary, and tertiary structure. And this is the primary strand of amino acids you see here. We'll go over this in class in a lot, maybe not much more detail on this, but each of these amino acids has a specific interaction with all of the other ones around it, which will give us possible shapes. One would be an alpha helical barrel shape here, or a beta pleated sheet here, where you can see the charges between them actually are repelling and attracting everything around them. What you need to understand is the sum of these charges, okay, will make what we hold is our tertiary structure. And to hold that tertiary structure together, we'll utilize hydrogen bonds. So I'm just going to put hydrogen next to this here. It's not the only type of bonding we use. We use sulfhydro bonding, stuff like that. But what happens if you need multiples of tertiary-based structures? Then you make what's called a quaternary structure, okay, where you have two of these. They look like they made mirror images of one another, and now they're functional, but they're all being held together pretty much the same way. Not pretty much. They are being held together the same way. You have a primary structure. They created secondary structures that are being held together in their correct tertiary structure for them to be functional, but because we required multiples, they're now being held together in their quaternary structure. So why do we care? Well, imagine, if you will, we go all the way back to how chemical reactions work, right? Let me go here. Chemical reactions work, and let me draw this for you, see if you remember it. Looks something like that, where we have our A plus B to give us our AB over here. And remember, we had our energy of activation. It came all the way over here. And then if we had a catalyst, and remember I said catalysts are inorganic, but enzymes are organic catalysts, we can lower the energy of activation here. Okay? And that means that an enzyme is a protein, and that protein has to be in its correct tertiary shape to allow for it to speed up chemical reactions. Okay? Which means if it's not in its correct conformational shape, it can't do this. And if it's not in its correct conformational shape, none of these things can happen here. Where an enzyme and a substrate come together, okay, it facilitates the reaction. You now make the products. They go off and do their own thing. And just like our catalyst, it doesn't harm the enzyme. This will continue to do the work it needs to do until such times as it's no longer needed. So, in the last couple of minutes here, what we're going to do is we're going to think about what are nucleic acids. And you're going to be surprised by some of our nucleic acids. Nucleic acids are huge organic molecules. They have carbon, hydrogen, oxygen, nitrogen, and notice now we're throwing in phosphorus. We did throw in some phosphate when we were looking at our phospholipids, but now we require that sugar-phosphate backbone. Deoxyribonucleic acid forms the genetic code inside each cell and thereby regulates the activity that takes place in that cell if those cells are with us for any period of time. Ribonucleic acid is an intermediate. It relays instructions from the genes inside the cell to now make specific proteins so that you can assemble amino acids correctly. Okay? Now, when we put these things together, this is what you need to understand. Here's a sugar-phosphate backbone on one side, a sugar-phosphate backbone on the other. We have 4 nucleotides that are held together by hydrogen bonding, which is completely correct. Okay? So think about it. All of your genetic information is being stored in a double helical structure that is alpha helical in nature, and it's holding together thymine, which binds to adenine, and cytosine, which binds to guanine. But these are our 4 nucleotides. Okay? And that's simply the information that's being stored. That information are the instructions now to make a specific RNA structure, which differs from DNA because it's single-stranded. It's a ribose sugar, not a deoxyribose sugar. And it has a uracil nitrogenous base instead of thymine. These are the rules. You need to make sure you understand it. It's just like a language. If you don't understand the language, there's no way you're going to make this work. Okay, now, there are different types. Okay, one is going to be made from DNA. Ribosomal and transfer RNAs, these are made from the nucleolus inside of the nucleus of a cell. If you don't know what that is, don't worry, we get to it inside chapter 3. But the significance here is you understand that by having different structures, now the cell knows these are completely different molecules. And because they're completely different molecules, they're going to have completely different jobs. The last nucleotide I want to introduce to you. Okay? That last nucleotide is adenosine, which is made up of adenine and a ribose sugar. And that ribose sugar has 3 phosphates attached to it if it's ATP. And the way this works is Mother Nature temporarily stores energy. Whoops, let's go like that. Let's go like that. Energy in between bonds. And the bond we usually think about is the terminal bond here. 1, 2, 3. And when you release that, when you cleave that, what happens is energy is released. And that energy allows us to excite electrons. And those excited electrons can change their conformation. They can now enter into chemical reactions. And now you can make new product, depending on what's needed. Okay, well, how does Mother Nature do this? Well, inside of your cells, there will be a mechanism by which you can hydrolyze ATP, and that's going to release that energy, which is going to now leave us our product of released energy and an ADP molecule. For us to now make new ATP, we will add a phosphate group. We will add a phosphate group to ADP. Inside your Bio 105 or your college biology class, you learn this can happen inside the cytosol of a cell during glycolysis, but most of it happens during the aerobic respiration phase inside of your mitochondria, okay, to give you 36 to 38 molecules of ATP. So we need our mitochondria to make ATP for us to utilize enzymes, so those enzymes can facilitate chemical reactions that are at a specific concentration inside of the liquid that makes up your body, which is plasma, which has ions traveling around inside of it. So that's a pretty good place to stop, and it's the end of chapter 2. So I'm kind of hoping you're going to come to class with questions, because we are going to finish this up and move into how do we build a cell now that we know how to use all these chemical reactions.